Welcome to a new and very strange world of nature. It's been taken over by the weird subatomic particles of quantum physics. As a physicist, I've spent my working life studying how these particles behave in the laboratory. But now I'm heading out into the natural world. I'm on a mission to prove that quantum physics can solve the greatest mysteries in biology.
This is a real adventure for me. I'm very much out of my comfort zone, trying to apply the very careful ideas I'm familiar with in a physics laboratory to the messy world of living things.
I believe that quantum physics could hold many of life's secrets. That deep in the cells of animals, particles glide through walls like ghosts. That when plants capture sunlight, their cells are invaded by shimmering waves that can be everywhere at the same time. And that even our human senses are tuning in to strange quantum vibrations. In the fantastic world of quantum biology, life is a game of chance, played by quantum rules.
This is what I hope to convince you of, to show you that quantum mechanics is essential in explaining many of the important processes in life, and potentially that quantum mechanics may even underpin the very existence of life itself.
My quest begins with one of the most majestic sights in nature: migration. Every winter, barnacled geese arrive right on cue at the same Scottish river. The end of an epic 2,000-mile voyage from Svalbard, high above the Arctic Circle. Of course, many birds head south for winter, then back home for summer. But for decades, exactly how birds navigated with such accuracy was one of the greatest mysteries in biology. So the most recent discovery has caused a sensation. In the past few years, one species of bird has helped create a scientific revolution. I was one of many physicists who were shocked to discover that it navigates using one of the strangest tricks in the whole of science. It utilizes a quirk of quantum mechanics, one that bamboozled even the greatest of physicists, from Richard Feynman to Albert Einstein himself. So you might be surprised to discover the identity of this mysterious creature. Say hello to the quantum robin. This is the European robin. Every year she migrates from northern Europe to the tip of Spain and back. In this laboratory in the woods, biologist Henrik Moritzen is trying to solve the mystery of how she does it. But he's found himself in my world, the strange world of quantum mechanics. Quantum mechanics describes the very weird behavior of subatomic particles. Down in this realm of the very small, we have to abandon common sense and intuition. Instead, this is a world where objects can spread out like waves. Quantum particles can be in many places at once and send each other mysterious communications.
I set out to understand how the bird finds its way, but it just turned out that the data more and more pointed towards this as the only explanation that could bring all the different results together.
Henrik's investigating a long-standing theory that robins navigate by the Earth's magnetic field. His laboratory is an ingenious magnetic birdcage. And these plastic cones, lined with scratch-sensitive paper, provide the key measurements. Henrik's artificial magnetic field is like the Earth's, except that he can point it in any direction he likes. Inside their cones, the robins always respond to the field, leaving scratches in a single direction. The big mystery is how. The Earth's magnetic field is incredibly weak, far too weak for any living creature to detect. But Henrik has found an intriguing clue by giving the quantum robin a mask.
We have a little leather hood similar to what you put on a falcon, you know, but just for a robin. And you have then a hole in front of one eye or a hole in front of the other eye. But what we can see is that if you cover up the right eye, you turn off the magnetic compass processing in the left part of the brain. If you cover up this eye, you turn the compass off in this part of the brain.
The robin's magnetic compass seems to be in her eyes.
I can show you what's going on using my own eye. Now, we use our eyes for vision, but we also have a second light detecting mechanism. If I shine this torch into my eye, you can see that my pupil closes down. It's basically a defense mechanism to protect my eyes.
My eye is responding to particles of light, or photons.
The energy provided by the photons is clearly enough to activate chemical reactions. After all, that's what controls my eye muscles.
Light must be causing similar chemical reactions in the robin's eyes. In fact, it's the power supply for a unique form of magnetic compass inside her cells, in the weird world of subatomic particles. A place where only quantum physics can explain what's going on. To see why, imagine the chemical reactions in the robin's eye taking place in mountains and valleys of energy. To get a reaction to start, you have to push molecules to the top of a mountain. Thanks to Henrik's experiments, we now know that light does most of the hard work. But when it reaches the very peak, the molecule becomes incredibly sensitive to the slightest touch.
The key point here is that the robin's chemical compass is now balanced on an energy peak between two valleys. Going one way produces one set of chemical products, the other a different set.
Now, even a tiny change in the Earth's magnetic field can tip the molecule over the top. But the way this happens defies common sense.
The final piece of the puzzle depends on one of the truly mind-boggling ideas in physics. But don't worry if you find it hard to understand. Even Albert Einstein called it spooky.
The idea is called quantum entanglement. It involves particles that seem to communicate faster than the speed of light. In 1935, Einstein published a famous paper arguing that it was impossible. But Einstein was wrong. In recent years, extremely delicate experiments have shown that subatomic particles really are entangled. It means they can subtly and instantaneously influence each other across space. And now it seems the same thing is going on inside the robin's eye.
When a photon enters the robin's eye, it creates what's called an entangled pair of electrons. Here's how it works. Each electron has two possible states. For simplicity, I'm choosing to call them red and green. Now, here's the weird thing. Until I measure it, it's neither one nor the other, but both at the same time.
Think of the electrons like spinning disks. They're simultaneously red and green. But by firing a dart, I can force the first electron to be one or the other. So far, it's just a game of chance. I don't know what I'll get until I try it.
So I know my first electron is red. Suppose I now measure the second electron. You'd think I'd have a 50-50 chance of getting red or green. After all, that's what you'd expect in the normal, everyday world. But you'd be wrong.
In quantum entanglement, the electrons are mysteriously linked. For example, if I get red on the first, I always get red on the second. It's not a game of chance anymore. It's as if the first electron is telling the second one what to do. That's why Einstein called it spooky.
The electrons seem to know that they should both have the same color, no matter how far apart they are.
The really important part is that the two electrons needn't be the same color. They can be entangled in a different way, so that if the first electron is red, the second one is always green. It seems that this mysterious connection is the ultimate secret of the quantum Robbins compass. Because the direction of the Earth's magnetic field can influence the outcome.
Near the equator, they may be more likely to be red-red, but near the pole, they may be more likely to be red-green. And that's the vital factor that finally tips the balance of the Robbins chemical compass.
Tiny variations in the Earth's magnetic field change the way electrons in the Robbins eye are entangled. And that's just enough to trigger her compass.
Now, finally, we can see how something as weak as the Earth's magnetic field can tip that balance one way or the other.
If the message changes, the chemical reaction tips a different way, changing the Robbins compass reading.
Suddenly it looks like it's a fundamentally quantum mechanical phenomenon in birds. It would be one of the first, if not the first, in biology.
Biologists better get used to the weirdness of physics. The robin is navigating by spooky quantum entanglement.
To see subtle quantum effects, even in a controlled, austere environment of a physics lab, is really difficult. And yet, here's the robin doing it with ease. These experiments are real and verifiable. And yet, even though I'm seeing them with my own eyes, I still find it hard to believe.
Bird navigation has brought physics and nature together as the science of quantum biology. There's a whole new world to explore. But its pioneers have found that it doesn't just affect birds. It affects every single one of us. Because the latest experiments say you're doing quantum physics right now. And believe it or not, you're doing it with your nose.
Hello, Jem. Hello. Hello, little girl. Hello. Our sense of smell is remarkable and quite different from our other senses of sight and hearing. Among the thousands of scents that we can recognize, many of them may well trigger very powerful memories and emotions. It's as though our sense of smell is wired directly to our inner consciousness. It's also different in another way. The other senses of sight and hearing rely on us detecting waves, light and sound. But our sense of smell involves detecting particles, chemical molecules. Recently, scientists have begun to realize that when it comes to our sense of smell, something very mysterious is going on.
For decades, biologists thought they knew exactly how our noses sniffed out different chemicals. But physicists like Jenny Brooks think there could be a new ingredient in the mix. And it smells like quantum mechanics.
A lot of people speak of the sense of smell and olfaction and the science of olfaction as being a problem that's been solved and we know all about it. And we do know a lot about it. We know about the ingredients, we know about the equipment that we use to smell. But I would argue that there's a little bit more to understand.
To understand more, I need someone to help me with a smell test. And Jem is going to sniff him out. Every human being gives off a cocktail of chemicals. Jem's nose could detect a single gram of it dissolved over an entire city. So she has no trouble finding the man I'm looking for. Meet Colin the gardener, a man who's used to smelling the flowers.
Right then, Colin, I'm going to put your sniffing skills to the test.
Cool.
I've got a selection of chemicals here, and I want you to tell me what they remind you of.
Okay.
I'll start you off easily.
Oh, that's like a minty vapor rub.
It is, yeah.
Sort of thing, like, yeah,
this is
sort of what you'd rub,
menthol.
menthol.
Yeah.
Yep. But it's that essence.
Right, here's the next one. So you should be able to recognise this one.
That's baking with my daughter. Mm-hmm. Icing sugar, sort of thing. Vanilla, yeah.
When our noses detect a chemical, they fire a nerve signal to our brains. But different chemicals create different sensations. The standard explanation for this is to do with the shape of the molecules.
The conventional theory that goes back to the 1950s says that the scent molecule has a particular shape that allows it to fit in to the receptor molecules in our nose. If it has the right shape, it's like a hand in a glove or a key in a lock. In fact, it's called the lock and key mechanism. With the wrong shape, it won't fit into the receptor. But with the right shape, it fits into the receptor, triggering that unique smell sensation.
Different receptors are wired to different parts of our brains. So when a menthol molecule locks into its specific receptor, it triggers that minty fresh sensation. But the lock and key theory has always had a problem. And Colin's next test will show you why.
Okay, how about this one? Quite strong smell.
Oh, that's yeah.
What does it remind you of? What does it conjure up? What memories?
I think Christmas cake.
Yeah, marzipan.
Yeah, that's it. Yeah,
Yes, almonds.
Very, yeah.
Colin identified the smell of marzipan or almonds. In fact, it's due to a scent molecule called benzaldehyde. What I didn't give him to smell was this other chemical, cyanide. Both benzaldehyde and cyanide have the same smell. They both smell of almonds. But these molecules are both very different shapes. So the lock and key mechanism as an explanation for how we smell can't be the whole story.
So why would two molecules with different shapes smell the same? Quantum biology has a head-spinning explanation. it says our noses aren't smelling chemical molecules. They're listening to them.
It's not just the shape of a scent molecule that matters. Let's take a closer look at this model of a cyanide molecule. The white ball here is a hydrogen atom, and the grey sticks are the bonds that hold it together with the carbon and nitrogen. But the reality isn't as simple as that. I can give you a better sense of what's going on if we look at this larger white ball. You see, atoms don't just sit still. The bonds that hold them together are like vibrating strings. And that gives us a whole new way of thinking about smell.
The bizarre new quantum theory of smell is all about vibrating bonds. Chemical molecules are playing music for our noses.
Imagine a receptor molecule in my nose is like my guitar. Before it can make a sound, a scent molecule has to enter my nose. And when that scent molecule is in place, its chemical bonds provide the strings, and it's ready to be played.
The receptor molecules contain quantum particles, electrons. As they leap from one atom to another, they vibrate the bonds of the scent molecule, like my fingers plucking a guitar string.
What's remarkable about this theory is that it tells us our sense of smell is about the vibrations of molecules, or wave-like behavior, and not so much about the shape of a particular scent molecule. Our sense of smell may be much more like our sense of hearing. A particular molecule, say that of grass, will vibrate at a particular frequency. But a different molecule, say that of mint, will vibrate at a different frequency.
This would explain why cyanide smells like almonds. The two molecules have different shapes, but their chemical bonds just happen to vibrate at the same frequency.
The quantum vibration in the odoron is almost literally like a particle of sound. So yeah, we're saying that the process of smell could be exactly like an acoustic resonance event. It could be very analogous to hearing and seeing, actually.
But can we really be listening with our noses? A bizarre theory needs a bizarre experiment to test it. Here's how it works. This molecule has a musky aroma, like perfume. But if the theory is right, then I should be able to change its smell by changing its vibrations.
The musky molecule contains lots of hydrogen atoms like this bonded to carbon atoms. But what if I were to replace all these atoms with a different form of hydrogen called deuterium? Now, it won't change the shape of the molecule, but it will change the way it vibrates. And here's why. Deuterium is twice as heavy as normal hydrogen, and so it vibrates more slowly. Now, different vibrations mean different smells. So if I were to make a new form of this chemical, all packed with deuterium atoms instead of normal hydrogen, it should smell different.
Quantum biologists found a unique way to carry out this experiment. A smell comparison using the most sensitive noses they could find. Fruit flies. First, the flies were trained to avoid the modified version of the musky molecule.
To be honest, I haven't got a clue how you go about training a fruit fly, but apparently you can.
In the laboratory, the flies had to pass through a kind of maze. They were then given a choice. Go right for the nice, musky smell, or left for the nasty, modified version. They could definitely smell the difference. They always preferred the original and turned right. The fruit fly experiment gives hard evidence that quantum smell theory really works. But ultimately, it works in harmony with the lock and key theory. First, the scent molecule fits into the receptor. Then those molecular vibrations take over. Incredible as it seems, flies, humans and dogs may be smelling the sound of quantum biology.
Our sense of smell is fascinating and mysterious as it is. But to think that when I encounter a particular scent and that sets off a whole wave of memories and emotions in my mind, that it's underpinned, that it's triggered by quantum mechanics, I think makes it even more remarkable.
The mysterious influence of quantum physics reaches into every corner of the natural world. In fact, it inhabits the walls of every living cell on Earth. Because the latest experiments suggest a magical solution to one of the greatest mysteries of nature. The miracle of metamorphosis. The transformation of a tadpole into a frog has never been fully explained. In little more than 6 weeks, the tadpole breaks down, then reassembles in its adult form. But the big mystery is how it happens so fast.
When you think about it, there's nothing more extraordinary than a tadpole turning into a frog. Take its tail, for example. Over a period of several weeks, it gets reabsorbed into the body, and the proteins and fibers that make up the flesh get recycled to form the frog's new limbs. But for this to happen, trillions and trillions of chemical reactions work together, breaking molecules, forming new ones in a carefully orchestrated dance. But the fibers that hold flesh together are very, very strong. They're a bit like these ropes holding my raft together. In order to dismantle the raft, I'd have to undo these very tight knots.
You can think of it like this. A tadpole is held together by long ropes of proteins, knotted together by chemical bonds. The bonds are so strong that they should last for years, much longer than the tadpole's entire lifespan. So how can it turn into a frog in just a few weeks? The explanation involves one of the most important molecules of life. Tiny widgets in all our cells called enzymes.
The enzymes are the actual machinery of the cell. They're actually the little machines inside cells that do the chemical transformations that are involved in everyday life. They're absolutely crucial. And the reason they're so crucial is because what they're able to do is to accelerate chemical reactions by enormous amounts.
Let me show you just how quickly enzymes get to work.
Inside this bottle is a substance called hydrogen peroxide. You're probably most familiar with it as the chemical used to bleach hair. In fact, I obtained this sample from my local hairdressers. Hydrogen peroxide is also produced in the body, and it's the job of the liver to get rid of it. Now, the way it does that is using an enzyme which breaks down hydrogen peroxide into water and oxygen. Now, to show you just how quickly this enzyme works, I'm going to do a quick demonstration. I've got some liver here which I've chopped up in order to release the enzyme. Now, watch what happens when I add this liver mixture containing the enzyme to the hydrogen peroxide. Watch how quickly the oxygen is released.
Just 100 grams of liver fired my rocket nearly 20 feet.
Liver enzymes make the breakdown of hydrogen peroxide incredibly efficient. It happens 1 trillion times faster. That's 1,000,000,000 times faster than it would otherwise.
In metamorphosis, it's enzymes that dismantle the tadpole's tail. And that means breaking down an incredibly tough protein called collagen.
Collagen is one of the most important proteins of the biological world. It's the protein which actually gives that resilience, that elasticity to tendons, to cartilage, and of course to our skin as well. And in the tail of the tadpole, it provides the kind of scaffold, right, that supports that structure. Now, when the tadpole is transformed into the frog, what you need to do is to essentially have an enzyme, collagenase, which will literally snip the collagen down into small pieces and thereby take that scaffold apart.
But how do enzymes break chemical bonds apart so incredibly fast? Let me show you why it's a problem only quantum biology can solve.
Think of it this way. All these different parts of the knot are like subatomic particles, electrons, protons, that hold the different parts of the molecule together. Now, to untie the knot, enzymes have to move protons about. But as you can see, this takes quite a bit of effort and a lot of time if there are many, many knots to unpick. Physicists have a fancy way of saying, put in effort to get something done. They say you have to overcome an energy barrier.
Okay, here's my energy barrier. And here's my proton. To break a bond apart, it needs enough energy to get over the barrier. The trouble is, when we work out how long this would take, it's much too slow to break down a tadpole's tail. But this is where protons turn into ghosts.
I wouldn't blame you for thinking that this is an idea that a clever theoretician has come up with, that it's just mere speculation, something that we have no proof of. But we do. It takes place all the time.
In the quantum world, protons don't have to go over barriers. They can tunnel straight through.
Tunneling strikes at the very heart of what is most strange about quantum mechanics. It's like nothing we see in our everyday world. A quantum particle can tunnel from one place to another, even if it has to pass through an impenetrable barrier. They're not solid objects like balls in our everyday world. They have spread out, fuzzy, wave-like behavior that allows them to leak through an energy barrier. A particle can disappear on one side of the barrier and instantaneously reappear on the other.
In nuclear physics, this effect is a proven fact. Without quantum tunneling, the sun simply wouldn't shine. But I never thought I'd see it in a tadpole.
It's hard to stress just how weird this process is. It's as though I would approach a solid brick wall and, like a phantom, disappear from one side and reappear on the other.
the most important advantage of tunneling is its speed. It happens incredibly quickly, much faster than if protons go over the barrier.
As a nuclear physicist, quantum tunneling is my bread and butter. Subatomic particles like protons do it all the time. But what has this got to do with biology?
The answer is that without quantum ghosts, the metamorphosis of a tadpole would be impossible. Remember, chemical bonds are basically knots. Tunneling unties them fast.
Have a look at these two knots. Now, on the face of it, they look identical, but there's a subtle difference. This knot has the two short ends of the rope on the same side, whereas this one has the two short ends on opposite sides. Now, you think that wouldn't make a difference, but it does. You see, this knot is very hard to break, whereas this one is easy.
Quantum tunneling turns strong knots into weak ones. So, in a tadpole, the entire collagen scaffold breaks apart easily. And finally, other enzymes rebuild it in the shape of a frog.
The quantum tunneling of particles is one of those weird features of the subatomic world that a physicist like me is very familiar with. After all, it's responsible for radioactive decay, and it goes on inside the sun. It's the reason why the sun and all stars shine. But to discover that it's going on inside every cell of every living organism on the planet, because every cell contains enzymes, now that I find truly amazing.
Quantum biology casts its spell over every living creature. We've seen that birds, mammals, insects, and amphibians are governed by the strangest laws in science. But the most dramatic recent breakthrough concerns the single vital process on which all these forms of life depend: the conversion of air and sunlight into plants.
This fine specimen is a Larix decidua, or European larch. It's about 100 feet high, and right at this moment, passing just this side of the planet Venus, is a bullet with this tree's name on it.
The bullet is a photon, nearing the end of its long journey from the sun. Its ultimate destiny is to kick-start a series of chemical reactions that underpins all life on Earth. Photosynthesis.
Every second of every day, 16,000 tons of new plant life are created on Earth. And for me, it's incredible to think that our existence on this planet depends on what happens in the next trillionth of a second.
The crucial first stage of photosynthesis is the capture of energy from the sun. It's nearly 100% efficient, vastly superior to any human technology. But the way that every plant on Earth achieves this is one of the great puzzles in biology. When it turned out that quantum weirdness might hold the answer, physicists could hardly believe it.
It was like a revelation. It was very exciting because I was used to work on problems that were quite abstract. Experiments, I'm a theoretician, but I always related my theory to experiments that were very clean in the lab, things that you can control. But now finding out that the things that I knew can help me to understand better how nature works, really, I don't know, scientifically, it was like a new inspiration to my life as a scientist. So I really, as I would say, I fell in love with this field.
Textbook biology says the color of green plants comes from chlorophyll molecules. Inside the living cells, they absorb light from the sun. This energy is then transferred incredibly quickly to the food-making factory at the heart of the cell. The entire event takes just a millionth of a second.
When the photon hits the cell, it knocks an electron out of the middle of a chlorophyll molecule. This creates a tiny packet of energy called an exciton. The exciton then bounces its way through a forest of chlorophyll molecules until it reaches what's called the reaction center. Now that's where its energy is used to drive chemical processes that create the all-important biomolecules of life. The problem is the exciton needs to find its way to the reaction center in the first place.
Textbook biology can't explain how the exciton does this. Because, of course, it doesn't know where it's going. It just bounces around like a pinball in a process called a random walk. Sooner or later, it'll pass through every single part of the cell. But this isn't the most efficient way to get around. Because when the exciton eventually does reach the reaction center, it's by pure chance.
If the exciton just blindly and randomly hops between the chlorophyll molecules, it would take too long to reach the reaction center and would have lost its energy as waste heat. But it doesn't. Something very different must be going on.
The vital clue comes from recent experiments that stunned the world of science. Chemists fired lasers at plant cells to simulate the capture of light from the sun. They confirmed the exciton wasn't bouncing along a haphazard route through the cell.
This original understanding didn't explain what we were observing in the lab. So the mystery relies in, okay, so then what is the explanation for what we are observing in the lab?
The solution is that plants obey the most famous law in all of quantum mechanics: the uncertainty principle. It says you can never be certain that the exciton is in one specific place. Instead, it behaves like a quantum wave, smearing itself out across the cell. The exciton doesn't simply move from A to B. In a bizarre but very real sense, it's heading in every direction at the same time.
It's spreading itself out as a wave so that it can explore all possible routes simultaneously. This strikes at the very heart of what's so strange about quantum mechanics. The exciton wave isn't just going this way or that way. It's following all paths at the same time. That's what gives it such incredible efficiency.
The beauty of it is, if the exciton is trying every route to the reaction center at once, it's bound to find the fastest possible way to deliver its energy. It's hard to express how incredible this discovery seems to physicists like me. Biological cells are full of the random jiggling of billions of atoms and molecules. But somehow, excitons maintain their form as beautiful, perfect quantum waves, transporting the energy that guarantees life on Earth.
It opened a whole new scientific path for me. And I really enjoy the fact that to be able to understand fully what is happening there or in the plants, you have to interact with scientists that have completely different approaches, like biologists and chemists. But we all have to come together to actually understand what is the relevance of this. So for me, this is one of the most exciting parts of this field.
Real scientific experiments leave no doubt. The strange hand of quantum mechanics has shaped the entire living world.
It's not a surprise that you should find quantum tricks being used in biological systems. The reason is because they're better.
Quantum entanglement is normally seen in the tightly controlled conditions of the physics lab. But now we know that robins use it to navigate with extraordinary precision. Quantum vibrations mean our noses listen to chemicals, enhancing our perception of the world around us. The living cells of all animals depend on protons that vanish and reappear like ghosts, speeding up the vital processes of life. And photosynthesis reveals the big picture. A shimmering world where quantum waves capture the sun's energy in an instant.
Sometimes people say, but physicists have been looking for this for decades. Well, biology has had millions of years.
The ultra-modern science of quantum mechanics is an ancient fact of life.
For the end of my journey, I want to take these ideas to their logical conclusion. Of course, as a scientist, any speculations I have to be backed up by careful experiments. So I want to concoct a thought experiment that helps me to answer the biggest biological question I can think of. Does quantum physics play any role in the mechanism of evolution itself?
In 1859, Charles Darwin stunned the world with his theory of evolution by natural selection. He went on to explain the differences between humans and other apes. 150 years later, there's no doubt that Darwin's theory accounts for every living organism on land and sea. But I'd like to explore the latest extraordinary interpretation of his ideas. Could there be a quantum theory of evolution? Can quantum evolution explain how the snail got its shell?
The snails I'm used to seeing in my back garden tend to have rather bland, boring shells. So have a look at this beauty. The patterns on its shell very perfectly match the lines on the stem. It's called a banded snail, Sepia nemoralis. And the pattern isn't there by accident. Come and have a look at this.
Less well-adapted snails are more likely to be found here.
This stone is called a thrush's anvil. The song thrush is the snail's main predator. It catches the snail and smashes its shell against the stone to get to the snail. Now, what I can see here is that there aren't many banded snail shells, suggesting that its colours camouflage it very well, hiding it from the bird.
Darwin's theory says that evolution depends on variation within a species. snails with camouflage are more likely to survive and reproduce, passing on their shells to the next generation so that the species as a whole becomes better adapted. So variation, the random differences between snails, is the driving force behind their evolution.
Now, all species evolve and adapt to their environment. But the question I'd like to explore is whether quantum mechanics plays a role in this.
The only way to find out is by scientific experiments. So my adventures in quantum biology finally bring me home to the University of Surrey. Here in the laboratories, I'm planning a new analysis of the most celebrated molecule in science. Deoxyribonucleic acid, or DNA. Its double helix holds the genetic code for every living organism.
It's a remarkable fact that Darwin himself had no idea what created variation in the species. The structure of DNA wasn't discovered until 1953 by Francis Crick and James Watson. The most famous feature of DNA is, of course, its beautiful double helix structure. But that's just scaffolding. The real genetic secret lies in between.
The 4 different colored molecules are called bases. The color code on one side, say blue, red, blue, forms a gene that parents pass on to their offspring.
A gene is a bit like a jigsaw puzzle. It fits together like this.
A full strand of the double helix forms a colored pattern. But the other strand always pairs up the same way. A blue base always goes with yellow, and green always goes with red. Because only those colors have the right shape to fit together.
What Crick and Watson realized was that this provides a mechanism for passing on the genetic code.
When cells reproduce, the 2 strands of DNA separate, ready to be copied. But red still goes with green, and yellow still goes with blue. So, bit by bit, the cell creates 2 new strands. 2 perfect copies of the entire genetic code.
So far, there's no genetic variation. This new copy is identical to the original. But here's the interesting bit. During the copying process, something very important can happen.
Sometimes mistakes creep in. They're called mutations.
Let's have a look at these 2 bases here. The 2 prongs that hold them together are subatomic particles. They're protons. They're basically the bonds between the strands of DNA. These protons can jump across to the other side. If the strands split when the protons have jumped across, they find themselves in the wrong position. Now, this red base will no longer bind to a green base. Instead, it has to bond to a yellow base. Slotting this back in, we see that now this copy is no longer identical to the original because I have a yellow base here instead of a green one. We've brought in a genetic mutation.
Jumping protons would change the snail's DNA. It could make a new gene for camouflage shells. The question is, how do protons jump? It's my belief that quantum spookiness can take over.
Now, for these mutations to take place, the protons have to overcome an energy barrier. And if you remember what happened with enzymes, well, you can probably guess what's coming next.
Protons can behave as if barriers don't exist. They tunnel straight through. But does this ghostly effect really happen? My colleagues in biology are already looking for the very first evidence of quantum mutations.
Biologists didn't really even know about quantum mechanics, so when you tell them that, you know, particles can be in two places at once, they kind of say, well, not in my cells, they can't.
Our experiment involves samples of bacteria. The first sample is prepared in normal water, containing hydrogen nuclei, or protons. When the bacteria reproduce, we simply count the mutations. But if our theory is correct, then we should be able to change the rate at which mutations occur.
Remember how we tested the quantum theory of smell? What if I replace the proton with its big brother, the deuteron? This is the nucleus of an atom of deuterium. Now, crucially, a deuteron is twice as heavy as a proton, and this should influence how easy it is for the deuteron to quantum tunnel.
Quantum mechanics is full of surprises. Protons tunnel easily. Deuterons don't. These heavier particles are much more likely to bounce straight back. So the second sample of bacteria is prepared in heavy water, which is full of deuterons. Our theory says you should get far fewer mutations. And so far, the results are extremely encouraging.
The preliminary experiments that we've done gives us a hint that the mutation rate is indeed depressed in deuterated water. We find that it is lower. So my hunch is that we're right, but we'll have to wait a little while before we're sure.
Final proof lies in the future. Even if we're right, quantum tunneling is a rare form of mutation. But our results promise hard evidence for a new explanation of one of the most fundamental processes of life.
Even the merest possibility of a new quantum mechanism for evolution itself is tremendously exciting. In fact, the story of quantum biology is only just beginning. What the frog, the robin, the fruit fly, and the tree have shown us is that real quantum effects are going on in nature all the time. And if there's anything we've learned from the history of quantum mechanics, it's this: we can never be certain where new discoveries will take us next.
Quantum biology is a revolution in science. But it's time I got back to the physics department.
Higher than the tallest building, higher than the biggest plane, James May goes to the edge of space. That's available now on BBC iPlayer. Next year on BBC4, though, we're uncovering the truth about meteors with Horizon.